Okay, so this is going to be chapter 8, infectious disease. And so when we take a look at infectious disease, we're basically looking at infections essentially of different things, so bacteria, viruses, and things like that. So before we get into it too deeply, we need to sort of describe exactly what we're talking about. So we have basically our microbes, which are essentially microscopic organisms. So for instance, bacteria, viruses, protists, we need a microscope to see them. They're all the way throughout our environment, so they're everywhere, um, and they're on all surfaces. Now, the problem is they get a bad rap because typically the ones that make the headlines are the bad ones that make us sick, but there are many of them that are actually quite useful to us. For instance, we use bacteria to make yogurt. We use bacteria to make cheese, bread, beer, wine. I mean, a lot of pickled foods have bacteria as part of the process. Some drugs are produced by bacteria, so they're helpful that way. And many of the bacteria are decomposers. Basically, all the leaf litter and things of that nature can get broken down by bacteria. So it's really important. They play a really important role in the environment. Now, those are the good ones. And by far, there's more good ones than bad ones. But the bad ones, are really bad. They basically make us sick and they cause disease and things of that nature. So generally speaking, we have to have our defense against the pathogens. For instance, we talked about that in chapter 7. So basically all the parts of our immune system, and I don't want to get too much into that because we covered an entire chapter of that. So our barriers, basically our skin, mucous membranes, our cells of our white blood cells, basically our phagocytic cells, which are doing battle with all these pathogens and killing them. And so they're also doing battle against abnormal cells like cancer cells and things like that. So we essentially have to sort of mount up a little bit of a defense against them. Now, let's take a look and see what exactly these things are. So, a bacterium is a very simple organism. It's a single-celled organism that is a prokaryote, which means it doesn't have a nucleus. And there are 3 major types of these typical organisms. We have based on shape. So we have the coccus, which are spherical shaped, bacillus, which typically are rod shaped. So they're kind of like little rods. Versus spheres. And we have spirillum, which are kind of like curved little corkscrew, squiggly little things that we oftentimes see. Most of them have a cell wall, and inside that cell wall is going to be a plasma membrane, which then contains all the plasma of the cell itself. But typically the cell wall is made out of something different. So for instance, as opposed to the cell wall of our plants, the bacterial cell walls are made out of a slightly different type of a thing that's called a peptidoglycan, which is essentially a kind of a carbohydrate protein combination molecule. So it's a disaccharide with an amino group on it. And so it's kind of like part protein, part carbohydrate. And so typically when we develop antibiotics like penicillin, for instance, they oftentimes will go target that cell wall because it is something that's unique to bacteria. So it's something that we can select for a particular antibacterial agent to go after. Oftentimes, they'll also be surrounded by a capsule, and this can have different consistencies. It can be hard or somewhat gooey or gelatinous. It'll allow these bacteria to actually stick to a hard surface, so they have some anchorage, and also helps to protect them as well. So this is kind of what it looks like. So going from the inside out, you can see in the middle of the bacterium, you're going to have what's called the nucleoid region. This is basically where you see your DNA piled up. And then outside of that is going to be your cytoplasm full of ribosomes, which are a protein manufacturing organelle. It's actually made of protein itself. And then on the outside, you'll see these membranes. So you first have your plasma membrane. And then the outside of that, you're going to see your cell wall. That's that peptidoglycan layer cell wall. And then on the outside of that, you're going to see your capsule for protection. And then typically bacteria will have all these little hairs on them called fimbriae. And these allows for like attachment and things of that nature. So they can attach to different things. And then oftentimes bacteria will have a long flagellum, and this is typically for locomotion. The other thing that I haven't mentioned as well is they have another source of DNA called a plasmid. It's basically like a little ring-like structure of DNA that it can use to kind of easily swap between bacteria. So bacteria don't really do sexual reproduction, so they're mostly asexual, but this plasmid allows them the ability to kind of swap different types of DNA material between different bacteria. So for instance, antibiotic resistance genes, for instance, can be easily swapped back and forth between bacteria in this manner. So then basically when we take a look at bacteria microbiology, we'll use a test called a Gram stain. And a Gram stain is basically just uses two different stains, and it takes advantage of the ability of the bacteria to differentially hold on to that stain. And so different bacteria will hold on to the stain differently, and so it drops them into two major groupings. So for instance, cell walls that have a thick peptide glycan layer tend to stain purple. And they retain that purple stain in their cell walls, and so oftentimes they're referred to as gram-positive bacteria with a Gram stain. And if they don't have a peptidoglycan layer in that cell wall, or it's a very small layer as well, what it'll do is it won't hold the purple, but it will hold the second stain, the second pink stain. And these ones will come across pink, and we'll call those gram-negative. And so generally speaking, these gram-negative bacteria will have an outer membrane, and typically what they'll have is these polysaccharides. So that's poly, many, saccharides means sugar. And so this is basically a lipid sugar kind of fusion molecule. And oftentimes when the cells are killed, they'll release these polysaccharides into the immune system, and then that'll create our inflammation and our fevers. So that's kind of part of what part of our infection symptomology. That's where that comes from. And so generally speaking, most bacteria will have a flagellum. As a matter of fact, we can classify them according to their flagella, but this is always to move the bacteria along. Some may or may not have fimbriae, which are basically stiff brush-like fibers. And this allows them to stick to surfaces, like for instance, our host cells. It's always a bad thing. But it also allows them to get access to our body. So they use their fimbriae as kind of a way to grab onto things and sort of stick to things, kind of like little Velcro. Little Velcro hairs. Oftentimes there's another type of fiber called the pilus or pili. And so these typically are a little bit longer than the fimbriae. They tend to have a hollowness to them, and their job is to transfer DNA from one cell to another. And so this basically will allow these two guys to communicate. So this is the process by which they swap these plasmid DNAs. So this is the process by which cells swap their plasmid DNA. Is through this conjugation and through what's called the conjugation pilus. Now DNA itself, which is in the prokaryote, is typically not packaged into a nucleus. It doesn't have a nucleus. It's just dumped into the center of the cell, and the center of that cell is called the nucleoid region. And then of course you have your little small plasmids, and this basically will have antibiotic resistance genes on it, and other genes as well that will allow you to be able to kind of swap information back and forth. So when we abuse antibiotics, basically we take antibiotics when we shouldn't be taking them. What it does is it basically increases the number of these resistant strains, and it makes it more and more difficult to kill the bacteria. So this is one of the reasons why we want to be judicious about our use of antibiotics, because when we do need them, we want them to work. We don't want to go back to the days before penicillin, because that was just a not very good time. So generally speaking, when bacteria reproduce, right, this is their reproduction strategy. They reproduce by what's called binary fission. So essentially they take one single circular chromosome, that is copied. And then these chromosomes are then separated to either ends of the cells. And then the cells themselves get separated into two cells. So the important thing to understand is that big chromosome that's in the middle of the nucleate region is still a big circle. It's just a really gigantic circle. So it just kind of collapses on itself and it's really big and floppy. And so generally speaking, E. coli, for instance, and at 37 degrees C will reproduce. Basically, it'll double every roughly 19 or so minutes. And it's kind of what it looks like. So basically here is, I'm going to use purple. So here is your two copied DNA circles. They've been copied. They've been sent to either end. And now what's happening is you're separating, that's what this little groove is right here. You're separating the cells into two. This is the fission piece, which means to separate. And so then you'll have two identical bacterial cells. Now, different types of bacteria that we run into, for instance, cause strep throat, tuberculosis, gangrene, gonorrhea, syphilis, a lot of bacterial diseases that are pretty bad. And so we have growth of bacteria, which starts to get in the way, but some bacteria will actually release toxins into your tissues, and that'll basically create a lot of problems. For instance, a good example is the tetanus bacterium, Clostridium tetani, or tetani. So this will basically produces a toxin that then interferes with the relaxation of muscles, creating constantly contracted muscles. That's tetanus, right? So when the muscles contract, they can't relax, and this is a problem. So it can cause suffocation if your diaphragm becomes contracted without relaxation. So you'll take a deep breath in, but you won't be able to breathe out, and you'll basically suffocate. Okay. Now, that's our bacteria. Now, what about viruses, right? This is another common thing that basically makes us all sick. So what about viruses? First of all, they're small. They're very parasitic. That means they can't live on their own. And they're non-living. So they're not actually a living thing. It means they're acellular. They're not a cell. They need a host cell to be able to reproduce. And there's only really two parts to a virus. They're very simple. There is a protein piece, typically called the capsid, which is like a little coat or jacket that surrounds the nucleic acid piece, which is the RNA or the DNA. This is the viral genome. It's kind of information that it needs to reproduce itself. And so this will basically carry its information for reproduction. So this is kind of what this relative size looks like. So here's a eukaryotic cell. This is us. Then here's a prokaryotic cell. This is a size of a bacterium. So you can see much, much smaller. And a virus is really, really tiny. So very, very tiny structures. Okay. So generally speaking, when you take a look at a virus, they're not cellular. The DNA, they're oftentimes segregated or classified by their genetic material. They're either DNA or an RNA genetic material. And so they're parasites, but they're parasites on cells. So they actually have to attack cells. They have to basically grab onto a host cell. And then what they do is they commandeer or they hijack all of the metabolic machinery of the host cell. And the host cell starts to do the work of the virus. And so that's basically what it does. That's what we were talking about before in the previous chapter about those virally infected cells. They'll start to make viral proteins. So the virus, the way it gains entry is that it requires a receptor. So it'll bind to a receptor on the host cell surface. Once it attaches, then it's going to insert its DNA or RNA, basically its genetic material, into the cell itself. And in that DNA is all the code for the proteins for the capsid and to basically rebuild new viruses. So it's basically like This is where we kind of get computer viruses from. So basically, they have the ability to commandeer your normal things, and it'll start using it for its own purposes. So some of the genes in the viral genome may encode for enzymes that are needed to make the viral products for reproduction, and some of them will be necessary for getting out of the host cell as well. So ultimately, the virus doesn't have anything except for the capsid proteins and its DNA. In order to do all this work, to make all of its particles for reproduction, it requires the host to supply the enzymes and the ribosomes, which are the protein factories, for its own reproduction. So it really does take advantage of all of the host work. So here's an example of a typical virus. So you can see you have your outer capsid portion, that's this little shell on the outside, and then on the inside you can see is your DNA genome, and that's pretty much all it has. So here's another different type of virus. So this is what's called an RNA virus. This is the flu virus. And so here you can see you have on the surface, you have this outer capsid region. That's the green piece in here. I'm going to switch to red here. So that's this green piece in here. That's your capsid. That's your protein capsid. This right here, the red part, is going to be your RNA genome. That's the genetic center of the virus. But then in addition to this, it's covered with what's called an envelope that surrounds it. So it's a second piece and a second outer layer of the virus. And in the envelope, you'll have what's called lots of spikes, which are basically proteins used to attach to your host cell. So a lot of the viruses that we study have these envelopes or enveloped viruses. And that's actually another way that you can classify viruses, is by their envelope as well. So one thing that we did in terms of understanding virology, so virology, which is the study of viruses in us, began with studying viruses in bacteria. So in bacteria, they have their own viruses as well. And that bacterial virus is called a bacteriophage. And so all of the basic components of a viral attack are exactly the same in this bacteriophage. And so we learned a lot about the life cycle of a virus and how viruses work by studying these bacteriophages. And so these are going to be important structures. First thing, in the life cycle of a virus, there's going to be attachment. The capsid of the virus itself has to attach to a receptor that's actually on the surface of the host cell. So there's an identification piece there. Once it gets attachment, then it moves on to penetration. This is basically where oftentimes an enzyme will digest part of the cell wall of the bacteria. In this case, it's bacteria. But we don't have cell walls, so you don't really need this particular piece associated with an animal viral attack because we don't have cell walls to get. We're a little easier to get into. But you have to chew away this peptidoglycan cell wall. And so there's an enzyme in there that will basically chew that up. Once it's open, then that genetic material is injected into the bacteria. And then ultimately, you have other viruses that can enter the cell as well. Then the third step, once your genetic material is inside the cell, it's biosynthesis time. Take over the machinery and use the host cell's own processes to replicate your viral proteins, your capsid proteins, your enzymes, and everything to reconstitute and to build new viruses inside the host cell. Now then, after that, from biosynthesis, you have to get to maturation. So maturation is essentially where you start to build your capsids and your genetic material. So you take the parts that have been made by the cell and build new virus. So now you got a host cell that's largely full of new viral particles. And then the last step is to release. So the new viruses will leave the cell, typically either by exocytosis or by bursting the cell membrane. And then that spews out all the new viruses. And so that's kind of the life cycle. And then you have a bunch of new viruses that can get basically kind of maraud around and infect new bacteria. Now, this is the basic bacteriophage virus. This is kind of the foundational pathway that we learned in bacteria, but there's lots of different alterations to this and a lot of different types of manipulations of this particular basic pathway. So here's kind of what it looks like. So starting off, first of all, with your attachment. So you're going to start off with your attachment. Your capsid has to basically bind to the receptor, and that's what's happening here. So here's your bacterial cell wall. You can see you're going to get some initial binding, recognition and binding of your host. Then what's going to happen is you're going to crack through that cell wall, and you're going to insert your viral DNA. That's penetration. From here, you have two different pathways to go. Either you can take your viral genome and insert it into the bacterial genome. That's a process called integration. If you integrate, you move into what's called the lysogenic style. So this is the cycle. This is like a dormant cycle, right? So ultimately what happens is you get to essentially make more copies of the virus and amplify the virus. So you basically get to increase the number of virus by normal cell growth. At this point, the bacteria is harboring this bacterial genome. And as this bacteria goes through its normal reproduction cycle and grows, then it's going to make copies of that viral genome as well. So this is one way to kind of increase the number of your viral genes. Now, you can also, from penetration, go directly to biosynthesis. So, or you can go from the lysogenic state, from the stored state, the dormant state, into biosynthesis. This is basically where you start to commandeer the mechanisms of the cell and you start to make little particles, so either genome pieces or capsid pieces. And so you're basically making the parts, making viral parts. And then from there, you're going to go to maturation, where you're going to start to assemble the virus. And then from there, you're going to distribute or release it. So oftentimes you're going to lyse the bacterial cell, which basically means you'll kill the bacterial cell, and then you'll have new virus, which then go find uninfected bacterial cells and basically infect even more. So a good example of this is the chickenpox virus, right? So the chickenpox virus typically will kind of go through its initial penetration stage. But what will happen a lot of times is it will integrate into the genome, and typically it integrates into the genome of nerve cells, and so it's in a lysogenic state. Why? Because at the end of life, at some point, what happens is those cells come out of dormancy and out of those nerve cells, and they start going to biosynthesis, and then that creates a lot of pain. That's shingles, right? So they kind of come out of their sleepfulness. So that's how viruses work. So in the lysogenic cycle, this is sort of like your dormant sort of building stage, where you're kind of just building up your viral army, if you will, just by using the cell's normal reproduction. And then you create this latent effect. So you'll get an infection, but it'll be latent. It will be down the road a little bit after you've been infected by the actual virus itself. So ultimately then the lysogenic stage is associated with the beginning of integration. And so typically what will happen is your viral DNA will incorporate into your host cell as what's called a prophage. And then it'll be replicated along with your host cell. Everything will be normal. These are lysogenic cells. They carry a copy of the viral information. And then ultimately what will happen is something will change, some sort of a environmental factor will occur, and then basically this will cause the prophage to enter into the lytic stage, and then you'll start to get the disease manifesting. So another interesting sort of an infection, which is a little bit different than viruses and bacteria, are what's called prions. These are basically something that's very unusual, which we don't usually see. These are basically what's called infectious proteins. So infectious protein particles are really unusual. It doesn't usually happen, but when they do happen, they typically will attack the nervous system. And they are the root cause of what's called Creutzfeldt-Jakob syndrome, which is essentially mad cow disease. So that's basically what these are called. This is the family of mad cow disease. In sheep, it's scrapie. In In cows, the mad cow disease is bovine spongiform encephalopathy. In humans, it's Creutzfeldt-Jakob disease. And so this is the same basic thing. It's caused by these infectious proteins. Normally what happens in a protein is they'll fold into an abnormal shape. And typically what happens is normal proteins will fold into a three-dimensional shape that makes them functional. But when they fold into an abnormal shape, they'll form into this kind of weird shape called a prion. And the prions are able to basically change the shape of other proteins. No other protein can do this. So as a prion comes into contact with another protein, it basically changes the shape of that protein into another prion. And that is the way it kind of is infectious, the way it basically infects other proteins. And then, of course, as you infect other proteins, it has a neurotoxic effect, which means your neurons start to die. And then that's when you have the mad cow syndrome. Okay. So let's take a look at some infectious disease. Typically, their classifications, we can classify infectious disease as either epidemic, which usually means that there are more cases than we expect in a certain particular area or period of time. And so if you have a rare disease that sort of pops up in numbers, we can refer to this as an epidemic as well. If you have a large number of a common disease, this may not necessarily be an epidemic. It just is a common disease, right? An outbreak typically is associated with an epidemic that is an increase in numbers located in a local area, right? So for instance, the Ebola outbreak of West Africa in 2013-14, right? So that was not a worldwide sort of a thing. That was just a local area where you had more cases of Ebola than normal. And so that became an epidemic in a localized area. So that was an outbreak. If the epidemic goes global, then we refer to it as a pandemic, right? So for instance, AIDS, HIV/AIDS, SARS-CoV-2, that's COVID-19, tuberculosis epidemics have gone global, so those become pandemics. Malaria, influenza, those are all current types of pandemics that just kind of stick around, and they're sort of a part of our normal global infectious disease backdrop. So an epidemic that goes global is a pandemic, okay? And this is kind of what it looks like in terms of outbreaks. So for instance, in an epidemic, you have one individual normally that you would expect. And so over time, you see multiple individuals more than you expect. An outbreak is basically the same idea, only it's in a local area. So you can see this is the same area. And then a pandemic, basically it goes global. So it's an epidemic, but then it starts to go global. So this is what they're trying to show here as a global spread. Okay. So now let's take a look at a couple of these individual ones. For instance, HIV/AIDS. We talked a little bit about this already, so I don't want to go through a lot of the stuff that we've already talked about. I want to talk about the specific things. So when we take a look at HIV, this is something that when we take a look at it, the first times we actually saw this, what we think was in the late 19th century to early 20th century. That's when we had the first human infection. Typically, we found it in human tissues, blood samples in the 50s and the 60s. And then the actual term AIDS came around in the early 80s. And that's usually when we referred to, when we kind of really started hearing about it across the world. And then, of course, we made the link between AIDS and HIV. HIV as a causative agent behind AIDS. So before they were separated, they were linked. They were not linked. We didn't know early on that AIDS was caused by HIV. That linkage didn't happen until more like the mid 80s. Now here's kind of some of the global statistics. I'm just going to go through this very quickly. So you can see the number of people that are living with HIV as opposed to the number of new infections versus the AIDS deaths. So you can see that in areas of Africa, you can see there's quite a few people that are living with HIV and quite a few new infections still happening. And of course, AIDS deaths. You can see it's a very strong problem in Africa and Asia and in the Pacific area. As we start to get into some of the European areas, for instance, those numbers go down. And so in the Middle East, they're down even more. So you can see there's a very different distribution. Africa is definitely suffering the most across the world. So here's basically the infections versus mortality. Now, basically in 1999, this is basically the dotted line is the number of HIV deaths happening in 1990, as opposed to the number of new HIV infections. So you can see everything kind of started off slow, but then as we go through the 90s, you can see you increased up to the early 2000s, the number of HIV-related deaths. That was kind of like a tail of the peak of the new cases, which happened in the mid to late 90s. So basically what you're seeing is it takes time to develop AIDS. So you don't develop it right away. Right, so there's kind of like this little bit of a delay right doing these two peaks. And now you can see that the number of new cases of HIV are down, and also the number of cases of deaths are down as well. So it appears as though we're starting to be able to kind of wrap our arms around the entire HIV scenario as well. Now let's take a look at the structure of the virus. The HIV virus is basically composed of two strands of RNA, so it's what's called a retrovirus. So any virus that has RNA as its genome is a retrovirus. It has several proteins, it has an envelope around it, and it acquires this envelope actually from its host cell. We'll see this here in just a little bit. The RNA is actually protected by 3 different protein coats. So it has a nucleocapsid, has a capsid, and it has a matrix. So it's very, very well layered, very well protected. So you can't really kill it very easily. And then in the matrix itself, you have 3 different enzymes. So basically reverse transcriptase is going to be allowing you to convert your RNA genome into DNA so that the host cell can use it. Then you have what's called integrase. This basically inserts the DNA into the host cell. So this is integration. And you have a protease. Basically, this is basically a protein cutter is what that is. It's an enzyme that essentially is a protein cutter. And its job is to activate new functional viral proteins. So here's kind of what the HIV structure looks like. So you can see in the middle, you've got your RNA here. And then you've got your integrase, which is the little red ball. You have the protease, which is the little blue ball, or the little blue square rather, and the reverse transcriptase, which is the little blue ball. And then from there, you're going to have this green layer, which is your capsid. And then you're going to have your matrix, which is that little pink layer. And then on the outside, you're going to have your envelope with all of these protein spikes in it. So you have multiple layers protecting your RNA genome. Okay. In the envelope itself, HIV has what's called GP120s or 120S. These are protein spikes that are in the envelope itself. And this is basically what it needs in order to get into target cells. So this is used for attachment. Has to have these. So they're very functional. Like I said before, this is a retrovirus. Basically, any virus that has an RNA genome instead of DNA is called a retrovirus, and as a result, they have to carry reverse transcription or reverse transcriptase, which allows you to convert your RNA into DNA. So those are some of the working parts of the HIV virus. Now let's take a look at its life cycle. So it first starts off just like the bacteriophages did with attachment. So it's going to bind to its target cell, and so the GP120 spikes will bind to what's called a CD4 receptor. So a CD4 receptor is the receptor on your T cells and your helper T cells. Remember in the last chapter, we talked a little bit about helper T cells having their own specific receptors. That receptor is what's called a CD4 receptor. And so it's unique to the helper T cells. So this is what the HIV will bind to. And then once it binds to the helper T cell, it's going to fuse with the plasma membrane. It's not going to insert, but it's going to basically fuse with the plasma membrane, and then everything inside the HIV is going to basically enter into the cell itself. And that basically gets us to entry, where you're going to have to enter the cell, but then in order to do that, you have to uncoat, right? So you have to take off your capsid. That's got to go. The other protein coats have got to go, right? Those are all removed, matrix, things like that, which releases the RNA and your viral proteins, those 3 viral proteins, where we have integrase, reverse transcriptase, and protease. You got to release those into the cytoplasm of the host cell so that you can do your job. So that's basically what's happening with entry. So ultimately, you're going to be releasing your reverse transcriptase, which typically is unique to retroviruses. They have to have that. And so reverse transcriptase will convert that single HIV RNA strand into a double-stranded viral DNA. And then from there, it will be ready for integration, right? So once you have your double-stranded DNA, then the integrase that came along with the HIV virus integrates that new viral DNA into the host cell, creating what's called a provirus. And so this is part now of the cell's genetic material. So this will expand with each cell division. So when your cell normally divides and basically goes through its activities, this provirus will come along with it. So you have the HIV virus now embedded in your own genome. So then what happens is, generally speaking, you transmit your HIV through the proviruses. That's typically the way you do that. And so the problem is, once it gets to the state, it's very difficult, right? Because even if you're able to detect these proviruses, which we refer to as viral load, or excuse me, other way around. If you're able to detect the viruses, this viral load, which is the free virus in your system, you still can't detect the provirus. This is hidden. From detection in our genome. So it's stuck in there. Then the next step is biosynthesis and cleavage. So once we kick into this stage, then we're going to take our normal cell machinery and we're going to start making more viral RNA. The RNA will code for different viral particles and polypeptides, and they'll be cut up into smaller pieces. That's cleavage. And then this is what's going to be activated by that protease, that HIV protease that's been hanging around. And so at this point, you're making new HIV virus. And then, of course, we get to the stage of assembly, which we saw before, where your capsid proteins, your viral enzymes, the RNA, these are all basically reassembled into new parts. And then you have what's called budding. This is a little bit different. So you don't lyse. So, but the new viruses leave the cell, but the way they leave the cell is by budding out. So here's your host cell. And instead of bursting like that, what happens is they bud out. So they basically kind of do this little thing like that. And so here's your new HIV virus. So what happens is the actual envelope is actually the host's plasma membrane. Right, so you basically now have this new HIV particle, which from the outside looks very much like the host plasma membrane. And what this does is it basically hides it from the immune system. Because you now look like your own cells. So here's kind of what it looks like. So your host DNA has your provirus embedded in it. It's going to be making all these different particles. So it's going to be making both the host genome and the protein particles. And then what's going to happen is your viral enzymes are essentially going to be making protein particles to rebuild your enzyme, right? But this is kind of a pictorial of the entire process. So you first start off by attachment. So you basically make contact between that GP120 protein spike and your CD4 receptor. Remember, this is a helper T cell. And then what happens is you're going to enter. So you're going to basically take your entire package. It's going to move in. You're going to break down those coats. Your viral DNA is going to be converted into, or your viral RNA is going to be converted into DNA. And that's going to enter into your nucleus, where you're going to integrate into your host DNA. And then from there, the provirus will activate and make all these different particles. So your viral mRNA is going to be made. So you're going to be making viral enzyme, capsid protein, and you're going to be making the spikes, right? So the spikes as well. So you can see here, you're making a bunch of spikes. And then what's going to happen is you're going to assemble all this stuff together. So it's biosynthesis and maturation. You're going to reform your coat around your viral genome, and you're basically going to move out to the periphery. You're going to bud out, but now what happens is you now look like this individual cell. So it's less likely that the immune system is going to be able to recognize this as a bad customer, because now you've cloaked yourself and disguised yourself looking like your own host cell. And so ultimately, when you transmit HIV to a new host, typically you're going to see this born in body secretions. Typically you'll have proviruses that will be hidden inside these helper T cells, right? These CD4 cells. So that's where the provirus stays. Where the provirus doesn't just go away. The provirus stays there in those cells. So they're terminally infected. There's no way to get rid of that at all. So ultimately, when these body secretions or any kind of an HIV infected secretion comes in contact with anything, so the vagina, mouth, infected cells migrate through the liner, blood, things like that, is essentially what happens is you have fluid on fluid contact. And then ultimately, you'll get contact between these two fluid sources, and you'll get HIV particles in contact with your tissues. In particular, the lining of the rectum is very, very thin. And so that's always a problem. So you can essentially transmit this, but it has to be through body fluids, right? So you can't sneeze it on somebody. You can't typically, say that there's a big issue in the 80s, right? So can I sit on a toilet? And no, you can't, right? It's got to be typically sexual contact or blood transfusions primarily is how it spreads, or how it's spread here in the US itself. So ultimately then these CD4 cells are typically the first ones infected, and the proviruses are in the body at this point. When they move to the lymph nodes, so ultimately what happens is they'll go to the T cells, the helper T cells. So it starts with the macrophages. Remember, the macrophages are the antigen presenting cells. And so when these guys will then go into the lymph nodes, then they will infect the CD4, the T cell, the helper T cells. And they can hide out here for a long period of time. But eventually they'll degenerate and you'll get large numbers of HIV particles in the bloodstream. Now, we before didn't have any solution for the HIV infection. Now we do. So we have a lot of different types of vaccinations. We have preventive vaccinations, which essentially helps us to prevent infection. Therapeutic vaccinations, which are essentially slows the progression of the disease for the future of infection, but it doesn't really solve the problem. And so we have a lot of different types of preventive vaccines, and a lot of different types of therapeutic vaccines. Typically, most of them are attacking some point of the life cycle of the HIV virus. So making it difficult for the HIV virus to sort of go through its life cycle. That's where most of these are coming from. And normally speaking, with somebody with HIV is taking a cocktail of different types of molecules and therapeutics to kind of hamper as much as possible the HIV virus. Tuberculosis. So this is another one. So this is oftentimes in the 19th century called consumption, because it felt like it kind of consumed you from the inside out, because oftentimes patients would spit up blood as they were coughing. And they felt like they essentially it was kind of a wasting away from the inside. We in the developing world in the West in particular had a lot of this in the 19th century. However, a lot of the world's population today is still exposed to tuberculosis. So a lot of those underdeveloped countries, that's the reason why we still have to keep our eyes open for it. And it's made by a type of bacteria called Mycobacterium tuberculosis. And so it's very, very difficult. You typically will spread it through droplets, for instance, introduced through air by coughing or singing or sneezing. And so this is always kind of a bit of a danger. So you got to be careful because they float in the air for a long, long time. So a lot of those cough droplets that we usually associate with people coughing or sneezing kind of hangs and lingers in the air for a long time. And so if we inhale those, then we can get ourselves infected. So typically it'll take a few weeks. So it'll take between a month to roughly 3 months or so for it to actually fully incubate before it starts to produce symptoms. So it develops very slowly. Which is very dangerous, right? Because it develops slowly, this basically allows a lot of time for spread, right? So you can easily spread this very quickly throughout a population because it is slow. A lot of times people won't even realize they have it, even though they'll be infectious. And so ultimately what happens is it's in the lungs. When it's in the lungs, the macrophages will go after it. And so other white blood cells will go there, and so they'll kind of wall off that area. And then what'll happen is they'll produce hard little nodules inside the lungs, or tubercles, which is why we call it tuberculosis. And that causes a problem, right? So this is what it looks like, the causative agent. This is that tuberculosis bacteria that you can see here. This is an electron micrograph. It's been colorized for us. But generally speaking, the bacteria will essentially hide within these hard tubercles, these hard nodules in your lungs. But the disease doesn't progress. So this is kind of like a late stage. And so ultimately this is a latent TB. So they oftentimes will not show symptoms. And typically they won't be contagious, but then they'll test positively for a TB test. And then once the tubercles calcify, they can be seen on a chest x-ray. So you can actually take a chest x-ray and see these tubercles. So for instance, if you take a look at this here, here's an infected lung. So you can see these little hard nodules in a person who's got hardened calcified tubercles from tuberculosis. So some of the symptoms of tuberculosis that we run into, basically, essentially what happens is the tubercle will ultimately liquefy, so it will become less hard. It will liquefy. It forms a little bit of a cavity, and from there, it'll liberate the bacteria, and then it can go spread to different cavities in the body, and it can be fatal, right? So ultimately, when you take a look at TB, usually it's you have a bad cough, you have chest pain, and you're coughing up blood, and those are pretty common signs. As a kid, going forward, you're going to get more tired, loss of appetite, chills, fevers, things like that. And then it starts to waste away. So patients start to lose weight. They start to waste away, which is a wasting sort of a thing. Okay. So tuberculosis has become a bit of a problem, even though we kind of had it beat pretty good there. Why? Because of antibiotic resistant strains, which essentially occurs because we're overusing antibiotics for things that we shouldn't be using antibiotics for. Like viral infections, for instance, don't respond to antibiotics, right? So ultimately then what happens is we have to give multiple antibiotic TB drugs for 12 to 24 months. So that's between a year and 2 years. So it's a constant bathing of antibiotics to be able to get a hold of these guys. And oftentimes we're starting to see now drug-resistant forms of TB, which are a problem. Because ultimately what happens is we have TB that we refer to as MDR, or multiple drug resistant TB, and another group called extensively drug resistant TB, XDR TB. Makes it harder and harder to kill. So these are TB strains that are becoming harder and harder to kill because now they're becoming resistant to antibiotics. Either way, it's going to take some time to kill off tuberculosis, at least a good 6 months. So it's a long-term chronic type of a treatment that you have to stick with it. Okay, so it's not an easy one. Malaria is another one, right? This is another one of our pandemics. So this is essentially a geographic distribution, mostly around the equator. typically delivered by mosquitoes as our main vector, which is essentially a living organism that carries the disease and transfers it to us. So that's basically what it does, transfers the pathogen from one host to another. That's a vector. It's a parasite. It's what's called a plasmodium parasite, which are protists. But ultimately, there are 4 species of these that actually infect human beings. Now, the important thing to understand is that when you're looking at malaria, that's mosquito bites, it's only the females that bite. And the reason is because they're incubating their eggs. So what will happen is the female will bite. She'll inject the saliva containing an anticoagulant to kind of thicken up the blood. But that comes along with the parasite. So she's injecting the parasite in you as well. And then the parasites will move through your system. They'll go into your liver where they affect your liver cells and your red blood cells. So you can have either mild malaria, flu-like illness, for instance, chills, fevers that just keeps recurring over and over again. But it can keep being cyclical. And so this is the reason why in subtropical areas, you'll see like mosquito nets. You'll see lots of anti-malarial drugs that you have to take, things of that nature to help prevent malaria. Can be quite deadly. Flu is the big killer, right? This is the one we oftentimes don't think of because we live with it, but it actually kills a lot of people, right? Up to almost a quarter of Americans, pretty much, will have the flu, and up to almost 40,000 deaths per year associated with the flu. So this is a viral infection, of course, has flu-like symptoms. We're all familiar with that, right? Runny nose, cough, chills, fever, head, things like that. Body aches, nausea. You can get this one by inhaling droplets, very similar to the same way you can get tuberculosis. The reason why you don't want to be around somebody who's coughing, right? You can also actually get it by touching hard objects with virus on it. So somebody's been like wiping their nose with their hands and they touch a tabletop, and then you come in after them and touch that tabletop, you've just picked up the virus. And when you smear it all over your face, you're basically getting that virus into your nasal cavity, and if you're infecting yourself. And so they basically will attack the respiratory tract for the most part. This is kind of what viruses do for a living. So here's kind of like another example. I know we had bird flu from time to time, but this is kind of what bird flu looks like. So this is a virus in birds. It has the ability to sort of jump over from one host to another, from birds to us. And so when you take a look at bird flu, you can see that you have your capsid, you have an envelope around it, you have your RNA genome. Typically what will happen is it'll go through one mutation that will change the nature of these little host cell identity spikes out there. And sometimes it'll go through another mutation, which will change another group of spikes. And so at this point, what happens is this will create then the ability for it to sort of evolve, if you will, and keep changing from year to year. So it kind of goes through these mutations and it changes the nature of these host cell spikes. This is basically how the flu virus works. This is the reason why we have to get a flu virus shot every year, because the flu virus keeps mutating into different forms and we have to kind of keep up with it. So one thing that can happen is sometimes we can actually combine viral genes and viral genomes. For instance, the human flu virus has the ability to sort of merge with the bird flu virus, creating a human centered combination of bird flu and human flu. And so we have a combination of both of those. And so viruses can do that. Those are very difficult to deal with. Okay. Now let's take a look at some of the emerging diseases. So I'm going to move through this section very quickly because this is like epidemiology. And then we'll finish up the chapter. So some of the emerging diseases that we have basically occurring for the first time in humans. They're happening more and more, and oftentimes they're geographic. For instance, avian flu, which is H5N1. So this is basically that those spike designation. Swine flu, which is H1N1. MERS, Zika, SARS-CoV-2, that's COVID-19. So these are emerging diseases. Re-emerging diseases typically reappear after a bit of a decline. For instance, outbreaks, if you will, of Streptococcus, Helicobacter pylori, things like that. So here you can see a map of the world with the different types of emerging diseases based on country. So you can see yellow fever, Ebola around the equator. You can see West Nile here in the Midwest, hantavirus in the Southwest, in Mexico, you can see H1N1. So you can see lots of different sort of spreads. There's a lot of different types of outbreaks of diseases across the world. So COVID-19 is basically named as a coronavirus. It's named after its crown-like spikes on its envelope. And so typically this is a pretty common type of virus. So COVID-19 is not the first time we ran into a coronavirus. So we have lots of animal viruses that are coronaviruses. We see them in bats, camels, birds, all sorts of stuff. We see them in ourselves. And so oftentimes we saw the first of the coronaviruses basically coming in the 50s, kind of created mild cold types of symptoms, but typically coronaviruses tend to be respiratory viruses. And in some cases, they tend to be intestinal viruses, and it depends on what host they're in. And so these are typically categorized into two large classes of coronaviruses. You either have the SARS group or the MERS group. And so those are the two main types of coronaviruses. In terms of the SARS-CoV coronaviruses, which CoV stands for, this is kind of the more severe type of coronavirus. So you have about a 10% fatality rate. And so we think that this came from bats. It was transmitted to humans, appeared in China for the first time in 2002, and then it spread to a global outbreak in 2003. And so that was the early version of this coronavirus outbreak. And so we had 8,000 cases roughly, about 700 or so deaths, so not anything close to COVID-19. MERS, then, the Middle Eastern Respiratory Syndrome, we saw basically moving. It's a coronavirus. And so we saw this basically moving across the Middle East. We didn't really know where it came from. I don't think we really know where it came from. We think it might be camels, maybe bats. But this is basically close contact between individuals, and it creates pneumonia and kidney failure. And it has a pretty significant mortality rate. So about a third of people who get it are likely going to die from MERS, which is the reason why MERS is such a really, really big thing for us to keep our eyes on. SARS-CoV-2 now is COVID-19. So this is the second version, the second coronavirus. The first one, SARS-CoV, came around in the early 2000s, and it wasn't as big of an outbreak. And then we have SARS-CoV-2, which is the COVID-19. And this was a new coronavirus, and we think it came from Wuhan in China because of some work with some bats there. Very contagious, became a global pandemic. All of us went through that in 2020. Millions of people were infected, millions of people died because of this. And so it disrupted society, caused society to shut down, created a lot of disaster, a lot of upheaval that we're still recovering from, to be quite honest with you. So let's take a look at antibiotic resistance. So basically, antibiotics use does not cause people to become resistant to drugs, right? It's pathogens themselves become resistant to drugs. And so typically speaking, there's going to be natural resistance in any population of bacteria. So when we use antibiotics to call the sensitive ones, then all that's left behind is the naturally resistant. So essentially, they acquire this resistance through mutations. That's one way to get it. And also through interactions with other organisms. They can swap it out with their fellow bacteria. The susceptible ones get killed by the antibiotics, but it leaves these naturally resistant ones to multiply. And this creates a new resistant population. And so that's a problem. That's how antibiotic resistance works. So if you basically take a look at this Petri dish, here's a Petri dish of bacteria. These two guys right here are naturally resistant. The rest of them are not. You expose them to the drug, it kills everybody off, with the exception of these two guys right here. Then what happens is these guys start to divide, and all of a sudden now your new population is now resistant. So notice your sensitive ones are much lower in number, and mostly what you have here are resistant. And that's how antibiotic resistance works. So ultimately, pretty much all of our diseases have acquired resistance: tuberculosis, malaria, gonorrhea, staphylococcus. A lot of these have, and so we have to be very careful in how we use antibiotics to make sure that we still have the ability to fight these off. But if we don't, there's going to be a day in the future where we're not going to have any medical treatment against these. And we're going to see rampant amounts of tuberculosis, malaria, and all sorts of stuff just basically running rampant through this population because our antibiotics will stop being effective because we overuse them. And so ultimately, some organisms are even moving from just individual antibiotic resistance to multi-drug resistance. And so some of these are leaving healthcare facilities and they're creating new infections. And so it's becoming difficult to manage. So ultimately, to prevent this, you have to take your antibiotics as directed. Don't overtake them, right? Don't skip doses, right? Don't discontinue when you feel better. Finish it out. There's a reason why we prescribe it as directed. We want to make sure that the particular cell is gone and that nobody's left behind to become resistant. Also, if it's a viral infection, don't take antibiotics for it. Colds, for instance, flus, for instance. Don't take antibiotics for it. I know it stinks to feel yucky, but that's the best way to do battle with the viral infection. And also, don't save unused antibiotics, right? Take only the ones that were prescribed because they're for different things. If you just start taking random old antibiotics, then you're going to be potentially creating resistance in your body. And so the number is increasing of the number of people getting infected with antibiotic resistant bacteria, right? So let's kind of circle back around to an idea that we mentioned, but we didn't really explain it. That's multi-drug resistance versus extensively drug resistant, right? So multi-drug resistant, or MDRO, comes in a couple of ways. So first of all, XDR, which we usually associate with TB, is basically when it has a resistance to pretty much almost all the drugs that's used to treat TB. This is dangerous because this basically means you're going to have untreatable TB coming for you pretty soon, right? So this is first-line antibiotics, the older, cheaper ones, right? As well as the second-line antibiotics, the newer, more expensive ones, right? So this basically is really bad. You don't want things to become XDR. And then you have the MDR, which is maybe just a lot of drugs, but not most of them, right? So there's different versions of this. So an example of this is methicillin-resistant Staphylococcus aureus. So basically it's resistance to methicillin, which is a penicillin derivative and other antibiotics. This is the one that causes staph infection, right? So in 1974, only 2% of staph infections were caused by MRSA. But by 2004, that has gone up to 63%. Why? Because it's resistant to antibiotics and it's starting to spread and it's becoming a problem. If you work in a hospital, staph infections are an enormous problem for hospitals. So you have to be careful. Okay, so that is chapter 8.